All right, welcome back. So this is the outro part of the dimensionality reduction lecture. And what we'll be talking about here is how we've been using dimensionality reduction in our research. And one of the questions that people often ask is dimensionality reduction good for anything beyond visualizing your neural activity? And the answer is yes. And what I hope to do here today is to show examples from our research where we've used dimensionality reduction in a closed-loop experiment to reveal something about the brain that would have been difficult to reveal otherwise. Okay, so before I dive in, I'd just like to acknowledge those who led this work. So Matthew Golub, Emily Obie, Patrick Sadler, who were or are current PhD students and postdocs. And they are all co-advised by Professors Aaron Batista, Steve Chase, and me. When you hear the term brain-computer interfaces, the first thing you probably think of is a paralyzed person controlling a robotic limb. And this is an important use of brain-computer interfaces. But there's another use of brain-computer interfaces which has not received as much attention, which I think is as important. And this is the use of brain-computer interface as a tool to study how the brain works. So a central message of this talk is that a brain-computer interface is a powerful tool for studying sensorimotor control. And so why might it do so? So it simplifies the brain's output interface without simplifying away the complexities of brain processing that we seek to understand. Okay, let's talk a little bit more about this. Okay, so let's talk about arm reaching. Okay, so here the effector is the arm. Okay, there are millions upon millions of non-output neurons. Right? These are essentially all the neurons in the brain. The number of output neurons are, say, the neurons projecting down the spinal cord, and there are many thousands of those. And in any given experiment, only a small number of them are typically recorded at the same time. Furthermore, the causal mapping between neuronal activity and the movements that are produced is typically unknown. And furthermore, the effector dynamics of the arm are difficult to measure and nonlinear, right? Because muscles have nonlinear dynamics, the arms have joints, etc. And furthermore, the sensory feedback, in particular proprioceptive feedback, is tied to the arm. So what we propose is to use a brain-computer interface to simplify matters. So a brain-computer interface is a system in which we can record neural activity from a population of neurons, and then translate that activity using BCI mapping into movements of a cursor on a computer screen. And this mapping takes the form of this equation that you see here, where x sub t is a 2x1 vector. That is the horizontal and vertical velocities of the BCI cursor. Xt minus 1 is the velocity of the cursor at the previous time point. U sub t is the vector of neural activity, of spike counts. So if we're recording from 100 neurons, the U sub t is a vector that's 100 by 1 and contains the counts of all 100 neurons, spike counts. And then big A, big B, and little b are the parameters of the BCI mapping. Okay, and so the subject's task is to move the cursor to hit targets, visual targets on a screen under moment-by-moment visual feedback. All right, so let's come back to this table. So under a BCI, the effector is now the BCI cursor or robotic limb. There are still millions upon millions of non-output neurons, right? These are still all the neurons in the brain. And one thing to realize here is, by the way in which we've set things up, we get to record from all of the output neurons by definition, right? And that only those neurons that we recorded can directly influence the movement of the BCI cursor or robotic limb. All the other neurons in the brain can still be involved, but in order for their activity to affect the BCI cursor or the robotic limb, they need to pass their signals through one or more of the neurons that we recorded. Okay. Furthermore, the neuron to movement mapping is known, right? That's the equation that we just talked about, right? We get to define that, and so we know it exactly. And furthermore, we can define that equation to be linear, which facilitates various analyses, and we'll see an example of this. And furthermore, the proprioceptive feedback is flexibly manipulable. We won't be talking about that today, but studies have leveraged this property to learn about proprioceptive feedback using BCIs. All right, so here are the key advantages of using a BCI. The first is that we can monitor all neurons that directly drive movement by the way in which we've set up the system. And this is extremely powerful because it's going to allow us to make scientifically causal statements about the relationship between neural activity that we record and behavior, meaning the cursor movements. Secondly, we can distinguish between output and non-output neurons. Okay, so this is relevant to questions about how brain areas communicate with each other. So just as a brain area only has a subset of the neurons that project to the next brain area, similarly, you can set up a BCI scenario in which you record from, say, 100 neurons, but only 50 of them are used to control the computer cursor, whereas the other 50 neurons are monitored passively. Okay? And we can study questions about how the activity of output neurons differs from non-output neurons. Furthermore, we can define a simple mapping, okay, like the linear equation that we talked about, and this facilitates various analyses of the data. Furthermore, we can change the BCI mapping during the experiment, and in doing so, we can study learning. And lastly, we can flexibly manipulate the proprioceptive feedback. Okay, and so in what we'll be talking about today, we'll be leveraging properties 1, 3, and 4 here. Okay, so learning requires the brain to alter the neural activity that it produces. Okay, so whether it be learning a sport, right, or some other physical activity, say dancing, or a more cognitive activity such as manipulating a Rubik's Cube or math or learning a new language. In all of these cases, learning requires the brain to alter the neural activity that it produces. Right? Between when you are a novice to when you become an expert, something changes in your brain. Okay, so we sought to address two fundamental questions. The first is, why are some tasks easier to learn than others? And the second is, how does neural population activity change during learning? Okay, so let's consider the first question. Okay, so why are some tasks easier to learn than others? Well, the first thing to realize is that different tasks require the brain to produce different patterns of neural activity. Okay, so what if it were the case that certain neural activity patterns were easier for the brain to produce than others? Right? That could be an explanation for why some tasks are easier to perform than others. And can we go as far as to ask whether subjects can learn to produce arbitrary population activity patterns? Okay, and if not, what defines the constraints on learning? Okay, so here is how we think about our neural activity. Okay, so we record from a population of tens of neurons. Okay, and in this cartoon, I'm just showing 3 neurons. Okay, and then we define a population activity space where each axis represents the activity of 1 neuron. We can define a window in time in which we count the number of spikes produced by each neuron, and that defines a point in this space. We can then define many such bins, right? And each bin corresponds to 1 point in this space. Okay, and an empirical observation with our BCI, okay, is we can define the BCI mapping in this space. Okay, so for example, if we define the BCI mapping this way, then if the subject wants to move the BCI cursor to the left, it will produce neural activity over here. And if the subject wants to move the BCI cursor to the right, it would need to produce neural activity up here. Okay. A key experimental manipulation, okay, in our experiments was that we switched the mapping unannounced to the subject. Okay. And what we asked was whether the subject could learn to use the new mapping. Okay, so now if the subject, to move the cursor to the left, the subject would need to produce neural activity up here. And for the subject to move the cursor to the right, it would need to produce neural activity down here. Okay. So as you would expect, right after we switched the mapping, the subject's performance was poor. Okay. And through trial and error, the subject might be able to learn to use this new mapping. Okay, so when we started these experiments, we switched the mapping on a few different sessions. And what we observed was that on for certain new mappings, the subject was able to learn to use it quite readily, whereas for other new mappings, the subject had difficulty learning to use the new mapping. So this led us to wonder whether there was some principle underlying which new mappings would be easier to learn and which new mappings would be more difficult to learn. So this led us to develop the following hypothesis, which is based on the empirical observation that these green points don't uniformly populate the entire population activity space. Rather, this neural activity tends to lie within some low-dimensional space, which we call the intrinsic manifold, within the population activity space. Okay, so we can think about this intrinsic manifold as reflecting underlying network constraints. Okay, meaning that, let's say, if neurons 1 and 2 are connected through an inhibitory connection, okay, one might expect that the activity of these 2 neurons to antivary. Okay, and that's reflected here, right, where the activity of neurons 1 and 2 antivary. So you can think about the intrinsic manifold as capturing all the pairwise correlation relationships among the neurons, right? And we, in the field, have been measuring these pairwise correlations for decades, and this low-dimensional space puts all these pairwise measurements into a coherent entity. Okay, so we developed the following hypothesis, okay, that we start with the BCI mapping, as shown here in black, and we can switch this BCI mapping in 2 ways. The first is to change the mapping in such a way such that the new mapping remains within the intrinsic manifold. And we predict that this would be easy for animals to learn. In contrast, if we switch the BCI mapping so that the new mapping is outside of the intrinsic manifold, that this should be harder for the subject to learn. Okay, so some of you might be thinking, wait a minute, isn't this obvious, why are you even wasting your time testing this? Right? So let me put two things on the table for you. Okay. The first is that a very important control in these experiments was that the initial difficulty of these two different types of new mappings were matched. Okay, so it's not as if right after we switched the mapping, this within manifold mappings were easy and outside manifold mappings were hard. Okay, that would not be an interesting experiment. Okay, rather, we want the initial difficulty right after we switch the mapping to be matched in the two cases, and then we can watch how learning proceeds from there. The second thing to put on the table is that previous studies have switched the BCI mapping, and even though they didn't call them outside manifold mappings, if you look back at how they switched the mapping, they were indeed of the outside manifold variety. And what they showed was that in some cases, animals were able to learn to use this outside manifold BCI mapping. So it's not as if this type of mapping is impossible for the animals to learn. Okay. So just to tell you a little bit more about our experiments, okay, we recorded using Utah arrays in motor cortex of macaque monkeys. We used threshold crossings, so there was no spike sorting. Spike counts were taken in non-overlapping 45 millisecond bins, and the animals performed an 8-target center out task. Okay, so let me show you data from a couple representative sessions. Okay, the vertical axis here is success rate, and the horizontal axis here is trial number throughout the course of a session. Okay, and here's a representative within manifold perturbation session. So at the beginning, the animal uses an intuitive mapping, okay, and performs at 100% with it. Okay, and at some point during the experiment, unannounced, we switch the mapping, and as you would expect, performance goes down. Okay, and through trial and error, the animal can learn to use this new mapping. Okay, and what you see here is that the animal gets back to near 100% correct. Okay, and at the end of the session, we then give the animal this intuitive mapping again. Okay, so you see here substantial learning. Okay, in contrast, here is an outside manifold perturbation session where again the animal starts off at near 100% correct. Okay, and then the mapping is switched, performance goes down. Okay, and through trial and error, performance improves a bit. Okay, and then it sort of peters out. Okay, and then we show the animal uses the intuitive mapping again. Okay, so the key takeaway here is that we see substantial learning here, and we see less learning here. Okay, so we did this experiment across tens of sessions in 2 animals. Okay, and what we saw was that we saw substantially more learning across within manifold perturbations than we did across outside manifold perturbations. Okay. So to summarize this first part, okay, what we found was that we saw substantially more learning within the intrinsic manifold than outside of the intrinsic manifold. And thus, the existing structure of a network can shape learning. Okay, so recall that the intrinsic manifold presumably reflects underlying network constraints about how neurons are interconnected with each other. And so the way in which neurons are wired up can affect what we find easier to learn or harder to learn. Okay, and now relating back to the theme of the day of dimensionality reduction, okay, what this is an example of is the use of dimensionality reduction in a closed loop experiment, okay, to reveal something about the brain that would have been difficult to reveal otherwise, which is causal constraints on the activity of population activity patterns attainable by networks of neurons. Okay. All right. So let's now move on to the second part, which is to ask, how does neural population activity change during learning? Okay, so for this, we're going to focus on the within manifold perturbation sessions, okay, because these are the sessions in which we saw the most learning. And we're going to look down into the intrinsic manifold and ask how neural activity reorganizes during learning. Okay, so here's the situation that we have before learning begins. Okay, so here's the BCI mapping, and when the animal wants to move the cursor to the left, it produces these green neural activity points, and when the animal wants to move the cursor to the right, it produces these purple activity points. Okay, now the mapping is switched unannounced to the animal, okay, and our key question now is, how does the animal reorganize its neural activity to regain control of the cursor using this perturbed mapping? All right, so what we did was to form various hypotheses about how neural activity can reorganize, and then ask which one of these possibilities was most consistent with the data. Okay, so let me go through each of these hypotheses in turn. The first hypothesis is called realignment. Okay, you can think about this as maximizing behavioral performance. Okay, so what happened here was that this green cloud, which used to be here, migrated up here, okay, to be consistent with moving the cursor to the left, and this purple cloud, which used to be here, migrated down here to be consistent with moving the cursor to the right. Okay, so this is consistent with moving the cursor very well according to this new mapping. And before we started this study, I would have put a lot of money on this hypothesis winning. Okay, the second hypothesis is called rescaling. Okay, you can think about this as restoring the influence of neural activity on behavior. Okay, so under the original mapping, neural activity changes along this horizontal axis would have had a large impact on cursor movements. Okay, but now with the mapping here, right, if you think about the projection of neural activity onto this mapping, changes in neural activity along the horizontal axis will only have a small effect on where the cursor goes. Okay, so under this hypothesis, neural activity scales up in this horizontal direction so that the variance of the projection of the activity onto this axis is the same as before. Okay, and so this is by analogy to visual motor gain adaptation. The third hypothesis is called reassociation. Okay, and all I've done here is to recolor the points, okay, where there are now more green points up here and then more purple points down here. Okay, and here the clouds did not move at all, right? All that's happened is that the animal would be using neural activity at different times, but similar neural activity patterns as before. Okay, we performed various tests on the real data, okay, and in each case, hypothesis 3 was a clear winner. Okay. So what this shows is that learning is not only constrained by an intrinsic manifold from part 1, right, it is further constrained by a neural repertoire, okay, meaning that over the course of 1 to 2 hours, it seems that the animal does not produce new neural activity patterns, but rather it uses its existing neural activity patterns at different times. Okay, and a corollary of this is that learning is suboptimal on the time scale of hours. Right? The optimal solution would have been hypothesis 1, which was not the winner. Okay, and since we did these experiments and the study, we've also asked whether giving the animal more time to learn could change the story. So Emily Obi, a postdoc with us, performed a heroic experiment in which she had animals learn to use outside manifold perturbations over the course of multiple days, up to 2 weeks. What she found was that learning over multiple days can overcome both types of constraints, both the intrinsic manifold constraint as well as the neural repertoire constraint. Okay, so now let's step back a bit, okay, and as we can see, BCIs enabled this work. Okay, so it did so by putting the task requirements directly on the neurons rather than on arm or eye movements, right? Which would have been putting the task requirements on the neurons only indirectly. And one of the strengths of using a BCI to study these questions is that we can causally attribute behavioral changes, i.e. movements of the cursor, to specific changes in the recorded activity. Right? Because we've recorded the entire set of output neurons, any changes that we see in the movements of the cursor, we can trace back to the activity of one or more of the neurons that we recorded. Right? And furthermore, because we define this neuron to movement mapping, so we know it exactly and we define it to be linear, it allowed us to define these 3 hypotheses in the second part concretely and test them. Okay, so I don't have time to get into some other studies that we've done, but I just like to mention them here, that we've also used BCIs to study internal models of sensorimotor control. And the idea of internal models is that when we move or when we interpret the world around us, we have some internal assumptions about how the world works. And we can use this BCI approach to identify what the subject's internal model is, and also how differences between the internal model and the way that world actually works can lead or explain why subjects make errors. We've also used BCI to study the question of neural redundancy. And so there are many neural activity patterns in the brain that can produce the same movement. How does the brain choose which of these many neural activity patterns is produced? Furthermore, we've studied this question of whether learning influences how we perform familiar tasks. Okay, so we have the animal did mapping A, mapping B, and mapping A again. What we asked was whether learning mapping B affects how you perform using mapping A. And what we do is to compare the activity in this second block of mapping A with the first block of mapping A. And what we found was that the activity in these 2 blocks is different, even though the animal is using the same mapping. And it was different in a way such that the activity in this block reflects a sort of memory trace of what the animal learned here. Okay, and then also we've been studying how the subject's engagement interacts with the learning process. Right, so you can imagine effects like arousal and attention that affect learning. Okay, and this study is a paper up on bioRxiv right now. Okay, so just to close, brain-computer interface is a powerful tool for studying sensorimotor control. Okay, because the BCI simplifies the brain's output interface without hopefully simplifying the complexity of brain processing that we seek to understand. Okay, BCIs engage many of the same cognitive processes, such as in arm reaching, such as learning, internal models, and more. Okay? And BCIs will hopefully be used to study other sensory, motor, and cognitive processes. And I think this is an exciting direction of future work. And I would expect that population analyses and dimensionality reduction methods would be essential in this endeavor, either as a central part of the experimental design, as what I showed in the first part here, or in the analyses of the neural activity, like what I showed in the second part here. Okay, so I'd just like to acknowledge my group, and also collaborators, and also our generous funding sources, and highlighted in red are all those people directly involved in the work discussed today. Thank you for your attention.